did my work in the Will Lab, and I assessed the impact of forest thinning treatments on terrestrial arthropod populations in the Santa Cruz Mountains. So for a bit of background, U.S. forest management began in around the 1870s with fire prevention campaigns centered around the goal of complete suppression of wildfires. But because of a lot of California's forests are adapted to frequent low-intensity fires and even thrive in their wake, forests became more dense and homogeneous as a result of this suppression. Due to a lot of overgrowth of plants, there was a lot of competition for resources in forests, leading to more stress and decreased resilience. This overgrowth also leads to an abundance of fuels, meaning these forests are more prone to severe and uncontrollable wildfires. Two new promising management methods for helping forests return to the resilience that they once had are controlled burning and mechanical thinning. However, mechanical thinning does have the potential to alter abiotic factors like microclimate and soil quality, which could have impacts on certain terrestrial arthropod groups that might be more sensitive to cover or climate changes. Now, why exactly do we care about arthropods? There's an important study done called the Fire and Fire Circuit study that looked at the effects of mechanical thinning and controlled burning on forests, and it found that they overall were very healthy for trees. And there was another study done by, I think, someone in that group, Makayla, who he looked at the terrestrial arthropod populations and how they changed. And he stated that ecological studies on their assemblages in California are very rare, meaning that our study is one of those studies. Arthropods in general are really, really important to their ecosystems. They're irreplaceable components in the food web, providers of essential nutrient cycling processes, and can serve as bioindicators for forest health. So our study was in the San Vicente Redwoods region. It's an area dominated by oak, Douglas fir, and madrone. We chose 9 plots that were minimally impacted by the 2020 CZU lightning complex fire in order to ensure proper results. If you chose ones that were too highly damaged, you would get proper thinning results. If they're too overgrown, you also wouldn't. So we chose a good middle ground. Each of these plots was treated with one of 3 mechanical thinning methods, high, medium, and low retention treatments. There were 3 plots of each of these treatment types. Us in the Will Lab, we buried 8 pitfall traps in each transect that were capped to a knot in use. And then during sampling, we're filled with 150 milliliters of propylene glycol, which is a mammal-safe agent that kills insects. The samples were taken in November and May, so we had a winter and spring season to it, over 2-week periods. These samples were collected after 1 week in order to prevent degradation. They were stored in refrigerated whirl packs, sorted by order, and then ID'd by students like myself. I personally compiled the data on the orthopterans, or the cricket species that were present. And my statistical analysis was done in RStudio using Formula to get some of my results. So my research questions were, does thinning of different intensities impact the population sizes and distribution of cricket and arthropod species across the plots? How might seasonal weather patterns impact cricket and arthropod abundance? And then how might these variables interact to affect abundance overall? I also had another question. Will the ant crickets that I found in my samples, will their abundance match the SVR ant population trends that were noted in a previous student, Mark Sun's thesis on ants and their responses to thinning? So here is one of the species present. Here are some pictures I took using the Will Labs camera, touched up a little in Photoshop. They are Christocithophilus pacificus. They're a camel cricket in the family Rhiphidophoridae. They're nocturnal generalists, but other than that, there's not a lot known about their phonology or ecology. They're often called nutcracker crickets because if you see that middle top picture, the males have a pretty crazy leg formation that they use to compete with other males and occasionally, unfortunately, to subdue female crickets. They're also defined by a prominent fastigial horn, which is basically like a little horn between the antennae. And the females have a serrated rather than a toothed ovipositor that can be seen on Pseudophilus. It looks kind of like the end of a butter knife. So special help to, or a thank you to Alice Avila and David Weitzman for helping me identify these and confirm my identifications, because they both stated these genera are in need of revision. Weissman also sent me a paper that listed as unnamed species that looks a lot like that striped individual that you can see on, I believe it's like my left, you guys' right, that might be an unnamed species. However, since I lack the taxonomic expertise right now to describe them, I'm putting them under P. pacificus for ease of analysis. We also have Makayla organensis. They're ant cricket species. They are obligate inquilines in ant nests. They're very, very small little guys, and they basically completely rely on ants for their lifestyles. My sample sizes of these guys were really small, which might have impacted some of the results I got, but they're still rather fascinating to look at anyways. So, my results for the Chrysostomphus, I found that there were significant increases between treatment years in the winter and significant decreases in the spring. I did not find that there was any significance related to treatment types in the winter, and there was a very slight significance in spring. And then I looked at both variables, treatment type and date or year combined, assessed with count, and there was still no significance for that. For the Myrmecophiles, there was a significant decrease between treatment years in the winter. Permanova said that that this p-value is insignificant for spring, even though there is a very big visual difference there. Again, no significance between treatment type or treatment type and date combined. I also did a combined analysis for any of the arthropod groups that were they had the data prepared during that time. It was some coleopterine groups, tenebrionids, carabids, crepuslionids, and then ant groups, formicids, and then my cricket data. So there were very highly significant decreases between the years, but not treatment type and not year and treatment type combined. So, in short, there seems to be little effect of actual treatment on the Orthoptera or on total arthropod abundance, as noted by the low significant values. So I believe that the changes, especially given that we only had one treatment year's worth of data, were likely due to environmental variables. I also found that looking at a gauge in the California-Nevada River Forecast Center's, there's precipitation data for the Ben Lomond gauge, which is very, very close to our study area. There was very high rainfall during winter of 2021 and spring of 2023, which is when we saw the big decreases in cricket populations. So I believe that may have been why they decreased so much. It supported my hypothesis partly that the crickets would decline as a result of seasonal differences and year differences, environmental differences, but not that they would decline in terms of treatment. However, they did see less of a decline and even an increase in winter than some of the other arthropod groups, so that part of my hypothesis was supported. Previous studies do seem to corroborate this difference, or seem to corroborate that there is more difference seen in arthropod assemblages during, as a result of environmental and seasonal value, or difference, let me slow down. As a result of environmental and seasonal variables rather than treatment, and overall they did find that treatment does increase heterogeneity and diversity among plots, and hopefully with the future of these SVR studies we will see that, and we will see that it has more positive impacts on arthropods. Few limitations, we only had one treatment year's worth of data that was available due to the really rigorous sorting process of the specimens. We had small sample sizes for the microphiles, and I think that in the future it might be best to do live capture studies to figure out which ants they cohabitate with, so that we could test the differences in those ant populations as opposed to relying on catching the myrmecophiles themselves. More research needs to be done on the cricket species that I've looked at in general. There was not a lot about their phenology, ecology, taxonomically they need revision. And also statistical analysis of the environmental variables, which we did not have time to extract from the tiny tag explorer that we used. Overall, it's clear that longer-term studies in the San Vicente redwoods are imperative to to properly assessing the effects of forest management. Forest thinning is a management practice on arthropod communities. However, diverse and healthy native arthropods are key to the functioning of many ecosystem processes, so their conservation should be highly considered when choosing management strategies. And that's it.
Yay.